Hello everyone, my name is Telmo Pérez. I'm a PhD student in the University of New Mexico, and thank you for listening on my work on recombinant intrinsically disordered proteins for trigger sequestration of nucleic acids via liquid-liquid phase separation. The idea behind this work is that we're capable of assembling and disassembling membraneless organelles composed of cationic intrinsically disordered proteins and single-stranded DNA. In order to achieve this, we induce the liquid-liquid phase separation of model IDPs known as elastin-like polypeptides that undergo liquid-liquid phase separation upon a thermal stimulus. So above a certain temperature named transition temperature or cloud point, we're capable of going from a soluble state to a two-phase regime in a fully reversible manner. And the two-phase regime is composed of protein poor and a protein-rich phase that is capable of sequestering nucleic acids and forming a membraneless-like organ. In order to achieve this, we use two elastin-like polypeptides, one with 8 lysines dispersed through the sequence named E3, and one with only 1 lysine named E140. We first studied the transition temperature or cloud point of E3 and E140 in the presence and absence of single-stranded DNA. We observed in panel A that the transition temperature or cloud point of this protein, the temperature at which they start phase separating, is altered in the presence of DNA for E3, but is not altered in the presence of single-stranded DNA for E140, which starts indicating us there is some type of interactions between E3 and DNA. By doing a fluorescence assay and a droplet-based temperature control fluorescence microscopy, panels B and C, we're capable of elucidating the presence of DNA in the protein-rich coacervate upon liquid-liquid phase separation. As you can see in image C, a site 3 label single-stranded DNA is distributed across this region of water oil droplets. Upon inducing liquid-liquid phase separation, you can see that there is a condensation of protein formed in the middle of the droplet, and that in this image, the DNA seems to be sequestered in these protein condensates, while if we incorporate some sodium chloride to shield the charges in E3 or with E140 that only has 1 lysine, it seems like the DNA doesn't prefer any phase or the other. It's distributed uniformly across the whole population of droplets, which is not the case for E3 without sodium chloride. And this process is reversible. So if we go back to 25 degrees, we can release the nucleic acid. And then we're going to have an on-demand capture of release and nucleic acids driven by liquid-liquid phase separation of ELPs, which are tunable phase separating agents that are highly modular. Right now, we're working on a theoretical framework that will define the component partitioning upon liquid-liquid phase separation and the engineering of new protein that incorporate robust nucleic acid binding domains to these liquid-liquid phase separating ELPs. Thank you for listening, and I'm waiting for your questions.